Another technique that we can use is we can produce a gene library. A gene library or a DNA library is a collection of clones that contain the entire genome of an organism. And so we're always trying to figure out what the DNA sequence is for an entire organism, and then what genes are produced. So we've done the Human Genome Project. We're always looking at the genomes of all organisms to see what proteins they produce and where those proteins are located, because that can help us to identify problems and also potential mechanisms for fixing problems in the body. And so to do this, we have to first isolate the genome from the organism that we care about. And so in this case, we're looking at bacteria X. Okay, so this could be any bacterium that we care about. We use restriction enzymes to generate fragments. Each fragment contains one specific gene. We then use those same restriction enzymes to cut up vectors. And then we allow the vectors to recombine with the restriction enzymes. We introduce the vectors into bacteria, and now we have a population of cells that contain fragments of the original genome. So each cell has a different fragment in it. We can do this with eukaryotes as well. But when we do this with eukaryotic organisms, we first have to get rid of the introns and produce mature messenger RNA. So from here, we can use a reverse transcriptase to reverse transcribe into a copy of DNA. From this, we can then use DNA polymerase to make double-stranded cDNA that we then can produce our own gene library from. So genes are produced by the sequence of DNA. And so knowing that sequence of DNA is really important. DNA sequencing is a mechanism to determine the order of nucleotides in your DNA. We've used this in the Human Genome Project, which allowed us to identify all the different genes in, well, obviously humans, but now we're using it to find genes in other organisms. So this has led to a field known as bioinformatics, which analyzes DNA, allows us to determine amino acid sequences, and then we can look at compare different proteins, and we can use this in the medical field if we're comparing proteins from an individual that has unique symptoms. We can look at the proteins of a normal person and then the proteins of this individual who is sick, and we can see if there's a change in that protein, if there's a mutation, and that can help us to identify certain illnesses. We can also use this to look at evolutionary relatedness to other organisms. So there are two methods: the dideoxy chain termination method, which is probably the most popular method, and this is also known as the Sanger method. And then there's a high throughput sequencing method. So we're going to talk about the Sanger method first. What the Sanger method uses is a DNA template. You have to have DNA polymerase. You have to have a primer, which researchers produce. So we determine where to start the synthesis. And then we have deoxynucleotides, normal DNA nucleotides, and then dideoxynucleotides, which are kind of like broken DNA. So once a dideoxynucleotide binds, nothing else can bind, okay? So once you have the dideoxynucleotides bind somewhere, then the chain stops. And what this does is it allows us to see where, say we produce a dideoxy A, okay, and I'm just going to put dideoxy, okay, and then we add these A's to the synthesis. So we have mainly regular A's, adenine, but we have some dideoxyadenine as well. So maybe 1/4 are this and 3/4 are this that we add. Now we're going to produce mostly this DNA right here, right? We're going to produce, we have the A's, we have the T's, the G's, and the C's, which are normal. And we're going to produce a lot of chains of the DNA, like just exactly what it's supposed to. But every once in a while, we're going to add in one of these dideoxys, and it stops DNA sequencing. What that's going to do is it's going to allow us to see where the A's are binding. So we do this in 4 different settings, okay? So you do this once, and then you have another group of Sanger technique, another Sanger techniques with the same DNA, but instead you use dideoxythymine. And in another group you use dideoxyguanine. And in another group you use dideoxycytosine. And what that allows us to do is find out where these C's are binding, where the guanine are binding, where the thymine are binding. And so that allows us to identify the sequence of DNA. This next technique, known as the high throughput sequencing method, is more automated. It's very fast. It doesn't cost as much. And it uses a lot of overlapping sequences of DNA. So the computers align and merge the DNA for analysis. And so down here you can see keep reading the textbook. Alignment, keep reading the textbook. The result, keep reading the textbook. So that's kind of how this technique works. But errors are very common, and so they have to be analyzed multiple times. There we go. So how does this work? So it's known as, we use nanopore sequencing, where we put long fragments of single-stranded DNA into an electrically conductive solution. We pass it through a microscopic pore, that's where that nanopore comes from, where different nucleotides block electric currents to varying degrees. And this allows us to know when an A or a T or a G or a C are located, and allows us to then figure out that sequence. I'm going to stop here, and we'll talk about the polymerase chain reaction in our next video. Bye.